This presentation will provide the calculations involved in marketing antibiotic susceptibility testing, otherwise referred to as AST systems. In order to evaluate any type of science, we need to have a way to measure the performance. There are numerous documents developed by organizations such as the FDA and CLSI, if you're in the United States, along with private and academic researchers. I will summarize some of those documents in this presentation. Before we jump into the exciting realm of AST device performance statistics, I'd like to cover some definitions of terminology I'll be using across the presentation that may be unfamiliar to some of the viewers. Now, minimum inhibitory concentrations are an endpoint measurement where the antibiotic concentrations that result in absence of bacterial growth, as indicated by turbidity, are reported. These values are discrete quantitative variables, meaning they are distinct numerical values. So we aren't measuring them, and we won't be evaluating a range of values that are uncountable. Antibiotic susceptibility testing is a test that measures the susceptibility of a bacterial species to antibiotics. Some examples of this includes gradient diffusion, such as the E-test and Lyophilchem MTS strips, macro and micro dilution panels, and automated systems that are commercially available, such as the Accelerate Pheno, bioMérieux Vitek 2, BD Phoenix, and some of the newer systems that are coming to the market, such as the Specific Reveal and Avails Medical EAST. Each system uses a standardized inoculum and introduces it to varying concentrations of antibiotics. The interpretations of the relationship between the bacteria and antibiotics are dependent upon which set of guidelines the user is intending to use, which is dependent on its use. So if we're using it in research or if we're using it in industry. Or region, so the United States versus the United Kingdom, and each uses their own separate interpretation of MIC results. Breakpoints are the concentrations of antibiotics used to describe the susceptibility or lack thereof of a bacterial species to a given antibiotic. And an isolate is a term I will use extensively, and it's a term used to refer to a single bacterial species. There are 6 descriptive statistics used to evaluate the performance of an AST device: essential agreement, a valuable essential agreement, categorical agreement, minor error, major errors, and very major errors. Essential agreement, or EA for short, is reporting the proportion of isolates that fall outside of 1-2-fold dilution from the reference MIC. So if your reference MIC is 2 mg per liter, then the test method must either be 1 mg per liter or 4 mg per liter to be within essential agreement. A valuable essential agreement, or EEA, is only calculated for MICs that have a discrete value. So any MICs containing less than or equal to or greater than or equal to signs are not considered for this statistic. Categorical agreement, or CA, is the agreement of interpretive results. This is monitoring if the test method matches the categorical bin of the reference method. In other words, are both methods calling a susceptible or a susceptible dose dependent or an intermediate or a resistant result? If the 2 methods match, then they are in categorical agreement. Minor errors, major errors, and very major errors fall within the categorical criteria. They are measuring the proportion of results that do not match at different extremes. As the name suggests, minor errors result in the smallest measurable error. Isolates fall within the minor error category if either the reference or test method results in either an intermediate or susceptible dose-dependent bin, and the other falls within either the susceptible or resistant bin. Major errors are when your reference method reports a susceptible result and your test method suggests it's resistant. A very major error is the flip of this, where the reference is resistant and the test is susceptible. These two can confuse people. Try to think about it from a clinical perspective. Both would be unethical, but one is worse than the other. A very major error could result in a clinician prescribing an antibiotic that is ineffective and result in clinical failure. A major error eliminates an effective therapy, so a medical professional would need to explore alternatives if the test was resistant. The slide here is showing the formula for each one of the statistics discussed on the previous slide. Each of these is reported as a proportion. The denominator contains the total number of isolates appropriate for that statistic, and the numerator of each is unique. In the numerator for the EA is the number of test MICs that are within 1 2-fold dilution of the reference. The numerator for the EEA is the same definition as EA, but the denominator now changes. The value is reported as the number of evaluable results. And as a reminder, this means it's absent of less than, equal to, or greater than signs. CA is the number of MICs that agree between the reference and test methods divided by the total number of organisms tested. Minor error is the number of minor errors divided by the total number of organisms. Major error is the number of major errors divided by the number of susceptible organisms determined by the reference. Very major error is the number of very major errors divided by the total number of resistant organisms by reference. Now, FDA suggests that manufacturers to target a 90% or above for CA and EA, and less than 3% for major errors, and 1.5% for very major errors. These are not definitive values, and each manufacturer should work directly with the FDA to determine the appropriate conditions for their technology to meet. Let's now look at an example. This slide is preparing you to evaluate an example of assessing results from a new AST device. The gold standard reference method is broth microdilution. The advantage of this method is it generates a quantitative result. Its reproducibility is great, and it's very cost effective. Some alternatives that one could explore includes macro dilution, gradient strips, disk diffusion, and automated systems. Our test method in this example is just a generic novel in vitro diagnostic device. During these analyses, you can look at one or more bacteria and drug combinations. Here we will look at one bacterium, E. coli, and one drug, meropenem, and we'll be evaluating a cohort of 100 E. coli. The table, which is a screenshot from the M100 CLSI document, shows the breakpoints for E. coli, which falls under the Enterobacterales order. MICs below than or equal to 1 mg per liter are considered susceptible, 2 mg per liter are intermediate, and greater than or equal to 4 mg per liter are resistant. There are a lot of ways you can go about presenting the results of the performance of an instrument. You could create a scattergram using computer languages such as Python or other statistical packages such as JMP. The example here just used Microsoft Excel. So the diagram on the left is showing the distribution of MICs captured by the reference and test methods. What this means is the same challenge isolate was tested on both methods, the broth microdilution being the reference, and the new in vitro diagnostic device serving as the test method. Best practice is to set up your MIC range for your test methods to match what your reference is. You can do this differently from the reference, but it makes the analysis more confusing. Underneath this diagram is the total number of evaluable results summed up for each of the available columns. I'll discuss this more later on in the presentation. In the table to the right below the organism are the summary results. We will go over each of these results one at a time in the coming slides. Starting with overall EA, we want to report the proportion of bacterial isolates that fall within 1 2-fold dilution from the reference. I'll have the formulas from earlier in this presentation displayed on each statistic we are looking at, so you don't have to flip between these slides and the original formula page. As the formula suggests, we want to count up all the organisms we tested and report this number in the denominator. Next, we want to tabulate the number of isolates that fell in essential agreement between the new test and the reference. To assist as a visual aid, I have included gray shading to show the isolates that fall within this agreement. Any numbers outside of these shaded areas are not in EA. If we sum up all the values outside of this gray region, we get 12. So we're going to subtract this from 100 to get 88. Our overall EA becomes 88 divided by 100, and then multiply this by 100 to get 88%. Notice we are including MICs that are reported with non-discrete MICs. The interpretation of this result is that 88% of the organisms tested were within 1 log 2 dilution when comparing the test method to the reference. Now you might be questioning why EA accounts for 1 twofold dilution in either direction of the reference MIC. To put it simply, it's because it's accounting for the innate variability that exists within broth microdilution testing. If you were to repeat a broth microdilution 10 times, you would most likely not get the exact same MIC every time. Occasionally it may drop below or above. Be careful with this though. If you start to notice that MIC is consistently dropping below or above, it's worth running quality control to determine if the biochemical properties of the media are changing, or if the drug is maybe degrading, or perhaps an organism has been subcultured too many times and has lost a resistance gene from its plasmid. These are just a few examples, and I just wanted to mention it so you have more tools to assess any erroneous data. On this slide, we're looking at EEA. So this is now looking at evaluable MICs, which again are those that do not have a less than or equal to sign or a greater than sign assigned to it. For EEA, we will only be including the values that fall within this red rectangle border on the diagram to the left. We won't be including the values outside, so we use a value of 94 for the denominator, and then approach the numerator in the same fashion as we did for EA. Sum up all the values in the gray region and use these as the numerator, so we get 86. So 86 divided by 94 is 0.915, and then we multiply this by 100 to get 91.5%. The interpretation of this result is that 91.5% of the organisms tested presented MICs that fell within 1 log 2 dilution when comparing the test method to the reference. Categorical agreement includes all of the isolates tested for the denominator. In the numerator, we are adding all the isolates that had matching categories between the reference and test methods. In other words, all the isolates that were susceptible in both methods, all the isolates that match the intermediate designation, and all the isolates captured as being resistant in the two methods. Any that didn't match are not in categorical agreement. It is very uncommon to see 100% CA. This can be explained by numerous variables such as human error, shifts in microbial phenotypic behavior. So researchers have actually found that certain bacteria such as Acinetobacter baumannii exhibit a phenomenon termed phenotypic switch, which can result in bacterial cells switching the appearance of the cell, such as white colonies to opaque colonies, or between avirulent to virulent. Other things to consider are inconsistent expression of antibiotic resistance genes, and a phenomenon termed as inoculum effect, and so much more. Coming back to the calculation of CA, of the 100 organisms evaluated, 60 were in categorical agreement. So we do 60 divided by 100 to get 0.6, and then we multiply this by 100 to get 60%. To interpret this, we say that 60% of the isolates tested were in categorical agreement across the reference and test methods. So this is the same thing. We're still looking at CA in this slide, but I've included some color overlays here to help you see which of the values belong in the numerator. All the isolates under the green are in agreement that the isolate is susceptible to meropenem. The 12 in the yellow agree that the species were intermediate, and the 12 in the red were in agreement the isolates were resistant to meropenem. So these are all the ones that we're adding up to get a value of 60. So again, we have 60% CA, and the interpretation of this is that 60% of the isolates tested were consistent in SIR, so susceptible, intermediate, resistant, between the methods that were compared. So within the categorical agreement umbrella, there are 3 primary values of importance when FDA looks at the performance of a device. These are minor errors, major errors, and very major errors. Minor errors are when the reference and test methods differ by one category. Let's say a reference was determined to be an intermediate MIC. A minor error would be when the test method is then either susceptible or it's reporting as resistant. In an instance such as evaluating daptomycin MICs when looking at Staphylococcus aureus, where there is no intermediate breakpoint value, you wouldn't be able to report minor errors. In that instance, you would either have major errors, so false non-susceptibility, or very major errors, false susceptibility. Try to keep in mind the statistic is only reported when you have an intermediary interpretive criteria. For minor errors, we're accounting for all organisms tested in the denominator, so 100 in this example, and the number of minor errors in the numerator. The red borders on the diagram show the values of interest. Any values inside of these are considered for the numerator. We can add up all the values and subtract out the 12 that agree the organism was found to be intermediate to meropenem, so we count 29, which results in 29 divided by 100, and then we multiply by 100 to get 29%. So to interpret this, we can say that 29% of the organisms tested were intermediate by one method and either susceptible or resistant by the other. Major errors occur when the testing method is unable to determine the true susceptibility of an organism and incorrectly reports a resistant result. We want to keep this rate below 3%. Referring to the example, we can use our formula located at the top of the slide to calculate the proportion of isolates that were incorrectly reported as resistant to meropenem when they should have been reported as susceptible. So looking at our table, we see there are 5 isolates that were found to be resistant at an MIC of 4 mg per liter by the test method, but susceptible by the reference method, and 1 isolate with an MIC that exceeded 4 mg per liter. So we have a total of 6 reported in the numerator of our fraction. To determine the denominator, we sum up all of the isolates with a susceptible result as indicated by the reference method, which is 42 in this example, and I've added in this red rectangular figure here to highlight which numbers are being included. So we get 6 divided by 42 and multiply this by 100 to get 14.2%. We can interpret this statistic by saying that approximately 14 to 15% of the isolates tested were incorrectly reported as resistant by the test method when they should have been susceptible to meropenem. This is actually a really high percentage, and I've rarely seen a major error reach this high, but it is possible. This gives researchers the opportunity to go back and optimize the assay. There are a lot of reasons to explain why the percentage may exceed 3%. To name some, it could be the biochemical properties of the assay, such as the glucose concentration or dissolved oxygen levels, inoculum effect, and variability in gene expression. I encourage people to explore doing repeat measurements of isolates to control some of these variables. If that still doesn't resolve the issue, it deserves a conversation to explore a different approach to implementing a test method. Now, the last thing I will mention is non-fermenting gram-negative bacteria are notorious for throwing off AST statistics. You may be able to get away with stratifying these results to perhaps remove these isolates from a panel if they continue to be problematic, or increase the number of repeat measurements you do and report modal MICs. The final AST statistic that I will go over is the very major error proportion. This statistic refers to the inability of the test method to detect resistance, so also known as false susceptible. Very major error may lead to the initiation of inadequate antimicrobial therapy and may have fatal consequences, especially in severely ill patients where these antibiotics are common first-line substances. On the other hand, major errors deprive the patients of treatment with an effective antibiotic and lead to the use of second-choice drugs, usually more recent and expensive, and thus contribute to economic losses in the selection of resistant strains. So let's keep in mind a very major error value should not exceed 1.5%. The equation to calculate this number is at the top of the slide, and I've moved over the red rectangle on the table to highlight the numbers of interest for this statistic. In the numerator, we are summing up the number of isolates that were reported as susceptible by the test method but found to be resistant by the reference method. So there are 5 of these in total, and we want to sum up all of the isolates within this red rectangle to fill in our denominator, so we get a total of 25. So to calculate the very major error, we would do 5 divided by 25 and multiply this by 100 to get 25%. To interpret the result, we can say that 25% of the isolates tested were false susceptible to meropenem as determined by the test method. Again, this number is very high and well above the 1.5% recommended cutoff value. The rationale for very major error being this high can be similar to the major error explanations, but I'll add some additional reasons here. So one thing to note is user differences. And so by this, I mean, depending on how someone was trained to interpret a result, they could incorrectly report a finding. In my experience, this occurs most frequently with broth microdilution manual testing and disk diffusion. And I want to revisit major errors for a moment because I just remembered this. If this were a true diagnostic device I was looking at, and I was getting these high major error rates, I'd look at the degradation of the antibiotic, which can be accomplished analytically using an HPLC or alternative instruments. Or, to be more cost effective, you can run quality control using ATCC organisms, and the protocol for this is discussed in detail in the CLSI documents. Something to note, just for this example, meropenem is much more stable than imipenem, and by that I mean it does not get hydrolyzed at the frequency that imipenem does. However, this does not exclude that meropenem can be hydrolyzed, or there could be some other underlying reason for why your meropenem is degrading. This concludes my presentation on the performance of AST devices. Please feel free to drop a comment below if there's anything I didn't answer or you're confused by, and like and/or subscribe to my channel for more discussions on microbiology topics. Thank you.